So here's a picture of a pig with a glowing nose, and this is not fake or doctored pictures. This is an actual pig. So if you held up a UV light to this pig, this is why it looks kind of yellow, because it's under a UV light. Its nose is going to glow, kind of like what we would see in a jellyfish or in like certain bugs, like lightning bugs or fireflies. So basically what's happened is that we have genetically engineered this pig to have a glowing nose and probably other glowing parts as well, but we've done this through DNA technology. We've been able to cut out a green fluorescent protein, or GFP, from a jellyfish, and we've inserted it into the pig's genome, or the pig's DNA, and it makes the pig start producing the GFP protein, and so that its nose starts to glow when we're looking at it. And so this is some of the technology that we have. And it can be a little bit scary to think about what we could actually do with it, but these are some of the trials that we have to actually use this biotechnology that we do have. So obviously you can think about some of the concerns and ethical issues with actually cutting and pasting DNA from different organisms together, or splicing it, because we could essentially get into what we think about as designer babies, and then we start coming into really, really, really ethical dilemmas. So genes as medicine. You can see the different things. We have glowing cats here. Obviously mama cat does not glow, but the kitten does. So again, using the GFP. Some of what we talk about as biotechnology is using genes as medicine. So we call that gene therapy. We have to look at the entire genome, so that's what this is doing here. And then we're also going to look at cloning. So one of the things that we can do, and this is used quite often in many, many, many different applications. We use it in forensics. We use it in DNA fingerprinting. We use it in determining paternity tests. So who's the baby daddy on Maury Povich, if you've ever watched that. So we use PCR, or polymerase chain reaction. So you remember what DNA polymerase does during DNA replication. It adds in all of the new nucleotide bases to the DNA strand. So using polymerase in this technique, basically we amplify, or we create a certain section of DNA, and we multiply it into millions and millions and millions of copies, so that we can actually look at and study a particular section of DNA from a very, very, very small amount. So essentially, if we have one tiny section of DNA, we can use this PCR technique to amplify or duplicate to make millions of copies of that same section of DNA, and then we can use it for lots of different things. We can study it to determine certain sequences in the DNA pattern. We can use it again for DNA fingerprinting. We can use it for paternity testing, and we can also use it to determine evolutionary relationships. So this process actually is the first step in many of the other types of biotechnology that we use. We have to go through this first. So what we need to know about PCR, polymerase chain reaction, we're using the enzyme DNA polymerase to make or duplicate into millions of copies a very small section of DNA. So why do we use PCR? Obviously we use it for DNA sequencing, so then I can look at and see what the normal DNA sequence should be for a certain section of a gene. But again, it is a common tool that's used in medical and biological research labs, and this is usually the first step of any kind of DNA technique or technology that I'm going to use. So we can look at and we can delete the presence or absence of a gene. So once we multiply it, we can basically delete a gene or a section of it, or we can insert some things in there and see what the outcome would be. So this is a way to kind of do this in a test tube in a lab setting before we actually do it in an organism to test if there's any ethical or safe issues that go along with it. We can identify pathogens during an infection, so if it's something that is altering the DNA, maybe specifically we're looking at like a viral infection here. And then we can generate samples. If we get a tiny, tiny, tiny sample of DNA from a crime scene, we can duplicate that millions of times so that we can definitely look at it and determine who the suspect is. If we know DNA from other suspects, we can compare them. So we all know what this is. This is a DNA double helix. You know everything about it because we've already gone over it. So everything you need to know, I'm not going to go over this in great detail again. So just know we have that sugar phosphate backbone, and then we have the complementary base pairs in the middle there. So this section that's highlighted right here, if this is a section of DNA that I'm interested in, I can essentially splice this out with restriction enzymes. We're going to talk about that in a second. But I can cut this out using restriction enzymes, which are like DNA scissors, and then I can copy that using PCR. So again, showing the double helix, this right here, again, a nucleotide. My phosphate of one sugar is bound to the sugar of the next nucleotide, so my sugar phosphate backbone with my complementary bases here. DNA changes, we've talked about those mutations, so I'm going to very briefly go into this. I don't know that these pictures are actually real pictures. They look like they're doctored a little bit, but these are definitely different types of flowers, and we can have lots of different types of mutations within the flower. So DNA can change a lot. It might also change a little, and it just depends on what is actually changed in the DNA to determine what type of effect we're going to see. Might be a big change, might be no change at all, it might be just a small change. So it just depends on what it is. Okay. And chromosomes, we know, are just super condensed DNA. They're wrapped around these histone proteins, and they coil all together so we can fit them all into the nucleus. So genes are just a certain segment of DNA that I'm interested in. So one gene codes for a single protein or a single trait. So all of your traits, again, are determined by proteins. And so a single gene determines a single protein or a single trait, and a gene is just a segment of DNA. On your chromosome, you have hundreds or thousands of different genes on each chromosome. So how big is human DNA? On average, it's about 5 centimeters long, and we have, this is called a karyotype. It is a map of all of your chromosomes. We'll look at those in our next unit. So we have 46 chromosomes in total, about 2 meters long, and all of that has to fit inside a tiny microscopic cell and inside an even tinier microscopic nucleus. All right, so restriction enzymes, and how do we actually use some of this technology to our benefit? Well, one of the most common things is the production of insulin. And so basically what we're doing here is that if we have this bacterial cell, this human pancreas cell, all the DNA in here, that means we have some sort of defective gene for creating insulin, which gives the patient diabetes. So we need to effectively produce insulin for them because their body cannot produce it. And so we know you can go, obviously you need a prescription, but you can go to the pharmacy and pick up your bottle of insulin. So it's not something we can just generate. We have to do it in a very specific pattern. So basically we have in your pancreas cell, we know what the gene should be to create insulin. So we have this human insulin producing gene. We can get a certain restriction enzyme, and so restriction enzymes are like DNA scissors, but they are very specific in that they know and recognize a particular sequence in the bases of DNA, and they cut that specific sequence out when they read it. So basically, if they're looking for a certain pattern of ATCG, that specific restriction enzyme will go along and it will read the DNA, and once it reads ATCG, it knows I have to cut this gene out, and it will do actually that. So these restriction enzymes were actually discovered when scientists were culturing bacteria. They were growing it on petri dishes, and they noticed that when they introduced viruses, some of the bacteria died, but some of it didn't. And so when they looked into it a little bit further, they realized that the bacteria that were surviving actually had these restriction enzymes that were specific to the viral DNA. So basically it would go cut up the viral DNA and it would protect the bacteria from that infection from the virus. And so basically it would stop the viral infection. So scientists have figured out how to use these to our benefit. So anyway, getting back to this, we have a very specific restriction enzyme that knows and can read the DNA to look for the gene that would make human insulin. So once we use that in a DNA strand from a human pancreas cell, it will cut out that gene. We can then take that gene here, and you can see it's this red piece here, and so we insert that gene that we've cut out using restriction enzymes. We insert it into the DNA of a bacteria. So remember, bacteria have a single chromosome and it's usually circular, and so we insert the gene for insulin into the bacterial DNA. And so when the bacteria's machinery, the ribosomes and everything, transcribe and translate its DNA, it's also transcribing and translating the gene for insulin. And so basically we get these bacteria to reproduce, and they pass along that gene as well. And so we get all these bacteria that are growing in culture that are mass producing insulin for us. So then we can just collect the insulin from the surrounding area, we put it into a vial, and then we can sell it at CVS for diabetes patients. So this is using restriction enzymes, and this is recombinant DNA technology. So think about, I'm recombining or combining the DNA of a couple of different organisms. Specifically, we're using bacteria to be able to do this. So bacteria are a great host or a great vector to be able to do this because they multiply so quickly that we get lots and lots and lots of bacteria with the gene that we want to transcribe and translate in a very short amount of time. And so we're able to mass produce insulin in a very, very short amount of time. And it's very effective for us, and it helps with a treatment. So restriction enzyme, they basically get their name from whatever organism they derive from. So one of the most common ones is EcoRI or EcoR1. And so it's isolated from E. coli. And so it recognizes the specific sequence you see here, GAA-TTC. And so once it reads a segment of DNA, it recognizes GAA-TTC, and it will cut out the entire segment. So both sides of the DNA will be cut out. So when do I actually use this PCR and restriction enzymes? So I can use it in DNA fingerprinting. And so DNA fingerprinting is basically matching up DNA that I find at a crime scene to a number of suspects. And so we know that DNA is specific to each person. It is different. And so just like our fingerprints are specific to each person, that's why we call this DNA fingerprinting. So basically, if I get some evidence at a crime scene, or even small amounts, which we call trace evidence, I can multiply it using PCR. Then I have enough to actually do this process. And once I do this process, I can match it up, and obviously it has to match up exactly, or else it is not our suspect. Because if it doesn't match exactly, that means it's DNA from a different person, because everybody's combination of DNA is different. So this process that I use in DNA fingerprinting is called gel electrophoresis. So basically I have this set up, and I create this gel. It's kind of like the agar we have in a Petri dish. And so once I use my PCR technique, I dye the DNA strand so that I can see it, and I put it in this gel plate, and it's got these tiny little wells at the top, so tiny little holes where I can put it in there. So the gel is inserted, so at these wells I put in my crime scene suspect DNA, and then I put in the DNA from suspect 1, 2, and 3, however many people I want to compare it to. And then I turn electricity on, and DNA has certain charges, so part of DNA is negative, part is positive, but most of the part on the outside is negatively charged. And so I have the electricity running, so positive end would be, whoops. So you can see the positive end would be up here, negative end would be down here. So once I turn the electricity on, we know that positive repels positive, negative repels negative. And so because of the repelling electrical properties coming from the electricity, and knowing that DNA is charged a little bit, it will push and separate all of the different segments of DNA that we have. And so it's separated on size. So it's kind of like going through this sponge. You can see in this sponge there's all different sizes of holes. Some are really big, some are really small. So if you were to push a molecule through this sponge, the bigger the molecule, the slower it's going to move, because it's probably not going to fit through these tiny, tiny, tiny holes very easily. Whereas small ones can pretty much pass all the way through really quickly without encountering too much friction to slow them down. So once I mix the DNA up with enzymes, certain restriction enzymes, I cut it up into smaller pieces, I place it into the gel, I turn on the power, and so it's moving towards the opposite sides. Then I kind of come out with this type